All right, there we go. All right, so let's start with the prayer real quick. In the name of the Father, and the Son, and the Holy Spirit. Amen. Our Father, who art in heaven, hallowed be thy name. Thy kingdom come, thy will be done on earth as it is in heaven. Give us this day our daily bread, and forgive us our trespasses, as we forgive those who trespass against us. And lead us not into temptation, but deliver us from evil. Amen. All right, other things that you should know. So for your exam next week, I had one question about this, and I'm guessing word has gotten around to you, but in case it hasn't, it will still cover chapter 7, 8, and 9. I am not adding chapter 10 onto that exam. So in case you were wondering about that, chapter 10 is not added onto the exam. All right, so chapter 10, 11, and if we get to it, 12 will be on the final exam. All right, only. All right, so to continue on today, so we're talking about membrane transport phenomena in chapter 10, and we left off talking about action potentials. Right, so and I talked to you guys about the root cause of action potential. So initially, there's basically the permeability of the membrane changes for sodium. Right, this causes an increase in the membrane potential, and then later on, the potassium channels open up. Right, and this causes a decrease in the membrane potential, and hence we get this this electrical response that occurs on the cell membrane. Right, and basically this electrical response will travel from one end of the neuron all the way down to the other end, and then to adjacent neurons, so on and so forth. All right, now what is actually causing these different channel opening and closing events? Right, well, for the channels opening and closing here, the sodium channels opening and then the potassium channels opening, these channels are gated by voltage. All right, so basically having a certain amount of charge across the charge difference across the membrane induces a conformational change in the protein. All right, now the question is, how does this happen? All right. So this is kind of a pictorial, cartoon picture of the potassium channel. So that's the one we're going to focus on. We're not going to worry about the sodium channel. All right, so the potassium channel, the voltage-gated potassium channel, has 6 transmembrane alpha helices. All right, and inside of the cell, there's a portion of the protein called an inactivation ball. And the most important parts of this protein, as far as understanding the mechanism by which it works, are alpha helical helix number 4, this transmembrane helix 4, and the inactivation ball. Those are the two parts that really cause this channel to fire. Now, this is depicted here on that alpha helix number 4. This alpha helix is special in that it is a very very positively charged alpha helix. So it has a lot of positively charged amino acids, even though it's located in the membrane portion of the protein. All right. So what does the channel actually look like? All right. So this is kind of on the left here. We have a top-down view of the channel. So you can see it's a homotetramer. There are 4 different subunits, the 4 different colors you see there. And in the center of the tetramer is a pore that is just wide enough for potassium to flow through. All right. This is now over here on the right. This is the side-on view. And you can see again that channel with the potassium ions as the green spheres, how they can basically flow through the membrane. The membrane would actually be, up in this region here. That's the membrane, that's the membrane. Show you how this protein would lie within the membrane structure. All right. Now, these potassium channels are going to have 2 different states to them. All right. They have an open state and a closed state. All right. So they're basically going to undergo a conformational change. Now, what is going to lead to that conformational change, and we'll see in a little bit this, is that alpha helix number 4, shown right here. And you can see, it's a little bit hard to see with this particular picture. Sorry, this is the picture the book gives. We've got an arginine here, another arginine, another arginine, another arginine. All right, so there are a lot of positively charged residues on this one particular alpha helix. All right, and what ends up happening is that between the open state and the closed state, so the open state is on the left, so the open over here, this is the closed state, is that positively charged alpha helix undergoes a conformational change. Basically, when it's in the open state, what is happening is that alpha helix bends into the upward position there. So it's kind of twisting and rotating upward. All right. This then basically opens a cavity in the central pore. Now, why would that alpha helix be moving upward? All right. What about this is causing that to occur? Well, when we're talking about membrane voltage, right, we're talking about differences in charges across the membrane, right? So normally when it's our helix is in this closed state here, all right, the voltages are relatively equal-ish across the membrane there. All right, but in order to open up this channel, what ends up happening is that the outside portion here becomes more negatively charged. The inside portion is becoming more positively charged. Or at least these various sides here. I'm not sure which side's inside and outside. Sorry. Basically, what ends up happening is that the positive charges on the alpha helix, they want to get away from the positive charge on one side of the membrane, and they're attracted towards the negative charges on the other side of the membrane. All right, so what is exactly causing this change is the charge differences on the sides of the membrane. So when we have a change in voltage, we're having a change in the distribution of the charges across the membrane. Basically, there's more positive charge on one side than the other, right? And that thereby is inducing this conformational change. All right, so that's it for our little foray into neurobiology. Neurobiology is a huge topic. I'm just giving you a brief preview of it. If you want to know it better, there are whole textbooks written on it. All right, which I've never read all that. All right. So our next type of transport, we're still in the passive transport area right now. We're going to talk about proteins called aquaporins. Right? Aquaporins are really crucial proteins that basically allow water to cross from one side of the cell membrane to another. So water is a relatively polar molecule, and so because of that, water can't just cross the cell membrane on its own very quickly. So for instance, if we had a change in the salinity outside of the cell, that's going to cause changes in that's going to. The cell is going to try to undergo osmosis, right, to basically re-equilibrate the water concentration on either side of the membrane. If there's no hole for water to get through, then basically the membrane is going to burst. Right? So these aquaporins are really critical just in order to allow exchange of water from one side of the membrane to another. Now, along with this, we know from introbiology classes that proton gradients, they're really important to life, right? So we talk about proton gradients with the mitochondria and how they have to basically pump protons out in order to get energy. All right, well, bacteria do similar sorts of things. So because of this, we want these pores to be able to let water through, but we don't want them to let protons through or base through. All right. So these pores are somehow able to do this. All right. Now, aquaporins, they form this tetramer-like cage here, and you can actually see the pore, here with the space filling model, the pore that the water will fit through. Here it's highlighting some of the amino acids that are in the pore structure. Here's the full cartoon. All right, but you can basically see there's a hole there it's a very narrow hole, but a hole nonetheless that molecules can flow through. All right, now how do these aquaporins actually work? Now, there a couple of different really key parts of this, right? So first of all, as I said, these pores, we don't want them to allow H3O plus through, right? We don't want them to let acid through. So because of that, or to prevent that, within the pore, there's this arginine residue and also a histidine next to it. These are both positively charged amino acids. All right, so these positively charged amino acids will tend to repel away any positively charged hydronium ions. All right, so as a result, those ions won't really have a means of getting through this channel. Now, water, though, is not quite that simple. It's not enough to just prevent hydronium from entering the channel. All right, the reason why, and we talked about this earlier in the class, is water has a unique ability to be able to form these proton hopping chains. All right, I'm going to draw one for you guys here again. So I'm going to draw a hydronium ion, H3O plus, and then a series of water molecules. All right, so in this example here, we're starting with the hydronium on the top, and we have three water molecules below it, right? All right, well, if that hydronium wanted to get all the way down to the bottom here, it could, maneuver its way past all these other water molecules and basically navigate its way over there. But that's going to take a relatively long amount of time compared to what we're going to talk about. All right, what we're talking about is a process called proton hopping. All right, well, what proton hopping is that it acknowledges the fact that the water molecule next to it has lone pairs of electrons that can react with and basically steal away that proton from the hydronium ion, right? So it can do that, steal that one away. Well, now the second water becomes a hydronium ion, but wait, this water down here can simultaneously do the same thing, right? It can also steal a proton from that one. Then the one below that could steal a proton from that one, right? So we have this entire chain where basically one oxygen of the water is able to steal a proton from an adjacent one, from an adjacent one, from an adjacent one, so on and so forth. All right. Well, the net process is that no atoms actually really moved any substantial distance, right? Yes, there's a slight movement of this hydrogen. This hydrogen, it moved ever so slightly downward towards that oxygen. This hydrogen moved ever so slightly downward towards that oxygen. So there's these tiny little movements. But in the end, the net reaction that was achieved is that the H3O plus that was up here is now an H3O plus down here. Right? And we achieved this just by basically doing all these little local proton hopping steps. All right, so that same sort of process could actually allow a proton to get all the way through this membrane. All right, so how does this channel actually prevent proton hopping across the membrane? Well, it does so with this one little special spot in here. All right, they call it, this water, it's essentially a water binding site in the middle of the channel. All right, now at that binding site, water is permitted in there, and let me draw it. That water loses its bonds to any other water molecules. Right? Instead, that water is entirely bound, hydrogen bound, to two different asparagines. I'm not to the side chains of these two asparagines that are there. Right? I guess I'll draw them as dotted lines rather than as actual bonds. So it forms hydrogen bonds. One single water molecule enters into there, forms hydrogen bonds with these two asparagines. Asparagines, the side chains of them, are they able to be protonated or deprotonated? We ever talked about an asparagine side chain getting protonated or deprotonated? No. So asparagine, right? The side chain of asparagine is a CH2. This is asparagine, right? We've never talked about that getting protonated or deprotonated, right? So basically asparagines, they're not able to act as an acid or a base, right? So because of this now, that water that's in the center of the cavity, its linkage and ability to do proton hopping chemistry has been broken, right? It is no longer bound at all to any other water molecules. The two molecules it is bound to are incapable of either stealing or donating protons. So because of that now, that entire hydrogen bonding chain or proton hopping chain that could have gone through the pore is now disrupted. Yeah, when I see some of these things in nature, just a little aside, I'm always amazed by it because like if you'd ask me to design, without knowing about this, you asked me to design a pore for water across the membrane, I would, design a pore, and I would have forgotten entirely about proton hopping. I would have had no idea how to prevent proton hopping, but somehow, nature evolved this ability. It's really pretty cool, in my opinion. All right, so that's our aquaporins. There's a few other pore types, or one or two, I think, in our passive transport portion of the chapter that I want to cover with you guys. This one is this is one of those little inset boxes in your chapter. These are things called gap junctions. All right, that's what we're going to talk about. So gap junctions are pores that physically link two cells together and actually allow for transport of items, different chemicals, directly from the inside of one cell to the inside of another cell without ever being outside the cell, right? So what we have here are basically two cells a cell on the left here, and then another cell on the right, right? And we have the two cell membranes, cell membrane 1, cell membrane 2, and this thing called a gap junction. It's a giant pore protein that is found in one of the membranes, and then that pore will form bonds basically with a similar pore in a adjacent cell. Now, the diameter of these pores is actually quite large. So lots and lots of different molecules can get through these gap junctions. So small things like ions, amino acids, sugars, right? Basically, now these two cells that are joined by a gap junction, let's say one of them just received sugar, the other one, that sugar molecule can be transported directly to the other cell, right? It also allows, this pore is big enough that proteins and nucleic acids can actually cross from one cell to another. Now, gap junctions are very, very common within biological tissues. So it's actually fair to say that biological tissues very often share one giant cell cytoplasm. Right? The cytosol basically of any one cell in your, let's say, muscle tissue or whatever, is actually linked via these gap junctions to all the other cytosols within that same tissue. All right, so this is how this really helps tissues function. So, for instance, if you're talking about a muscle tissue, all right, well, one of the muscle cells that is adjacent to a blood vessel, it's relatively easy for that cell to get glucose from the blood, right, or oxygen from the blood. A muscle cell that's further inward, all right, it's harder for it to get those materials. Well, because of gap junctions, the sugars that are received by a cell are right next to a blood vessel, basically any sugars or oxygen can then be transported directly to adjacent cells they're further or deeper into the tissue. All right, and this is just a actual X-ray structure showing you this here. And so these are relatively large protein complexes, and they have a big old pore in the center of them. All right, another passive transport system we're going to talk about. This is an example of a gated transporter that recognizes the molecule that's being transported. All right. So basically, if the molecule that is going to be transported through these transporters binds to the pore opens and allows it to pass through. Otherwise, it's closed. All right. So the example we're going to use here is a glucose transporter. So glucose transporters, I think the importance of them is pretty obvious. Why does the cell need glucose for energy, right? Now, the way that these transporters work is their native resting state is shown on the top left. All right, so usually these transporters are open to the outside of the cell, but there's essentially no path through this transporter. They're closed. They're always a closed pore. Right? These actually never fully open up. So you can see basically on the bottom, it's closed down there. The top part is open to the outside of the cell, but things can't just go through this. Right? Well, what ends up happening with the glucose transporter is the glucose molecule will enter into that open side and bind within the pore of this protein. Right? Once it is bound, the binding of glucose actually then triggers a conformational change in the protein. Right? The conformational change closes the protein on the top and opens it on the bottom. All right, so now that glucose is able to diffuse away to the other side of the cell. All right, so I would call this a ligand-gated passive transporter. So the ligand glucose is inducing a conformational change that causes itself to be transported from one side of the cell membrane to another. All right, this is what the GLUT1 protein, the glucose transporter, actually looks like. You can see here a pore allowing the glucose, which is shown in the green and red spheres there, to enter into that cavity and bind to it. All right, now, passive transport of glucose seems really nice in that you don't have to expend any energy to do this, right? The problem enters in for our bodies. We have to maintain a certain level of blood sugar, certain blood sugar levels, right? So we don't always want to be importing in glucose. We want to be able to basically control this process. Under certain conditions, our cells need more glucose. Under certain conditions, they need less. All right, so if we're talking about a membrane transporter that's out there and just simply responding to glucose levels, all right, that doesn't necessarily tell us how nature is able to control this process. All right, so if we're talking about these transporters, how can nature actually, increase the amount of transporters at a membrane or decrease them? Well, the way that this happens is that any transporters that we have on the cell surface can be removed from the cell surface by a process called endocytosis. Basically, the cell membrane can start to invaginate. There are a whole host of proteins involved in this process and triggers and stuff like that. But it starts to invaginate until it actually buds off, forming a vesicle inside of the cell. All right, that vesicle contains the transporters. They're still there. All right, they're just not on the outside of the cell, so they're not performing their function. In this case, glucose transport. All right, now, in our cells, so our glucose receptors, when we have kind of normal blood sugar levels, all right, there's going to be a certain number of glucose transporters that are basically brought into the cell because we don't need to worry so much about our glucose concentration. Now, if we have high blood glucose levels, all right, what's our body? Anyone know the hormonal response of our bodies to high glucose levels? What happens in response to high glucose levels? insulin gets excreted. All right, so basically, if there's high blood sugar, insulin levels go up. All right, so at that point now, our body's trying to basically pull glucose out of our blood. So we want our cells to be uptaking glucose because there's too much of it around. All right. Well, what insulin does, part of what insulin does, all right, is it causes these vesicles that contain the GLUT4 transporter to go and fuse with the cell membrane. All right, this process is called exocytosis. You know, we talked about how snare proteins can be involved in this, for instance, trying to cause the fusion of basically a vesicle membrane with the plasma membrane. So a lot of those same sorts of proteins that we've talked about previously will be involved in this process. But in the end now, those transporters are now back out at the cell surface, and they're able to transport glucose across. So, you know, cells. The take-home message here is that cells can respond to hormonal stimulation or different sorts of triggers to transport to remove transporters from the plasma membrane using endocytosis, or to put more back out at the plasma membrane using exocytosis. All right. Now, a couple other things here. In this, we're starting to transition away from passive transport towards active transport. But before we do so, this slide here really applies to any sort of a transporter. All right. So this is another terminology thing that's kind of in an awkward place in the slides. Sorry. So we can divide our transporters into active or passive transporters. You know, whether they rely simply on diffusion, or if they actually involve energy, or if they need energy to actually do the transport. That's the difference between passive and active. We can also alternatively divide our transporters into the way shown here. Right? We have uniporters, where basically one molecule is just simply brought across the membrane. So our potassium channel would be in a uniporter. The glucose transporter would be a uniporter. We also have symporters. Right? Symporters will take two different chemicals across the cell membrane, but the two chemicals are moving in the same direction. Right? We have not yet talked about a symporter. I think we cover one of them in the class. And then we also have examples of proteins called antiporters. Right? Antiporters basically will move two chemicals across the membrane, but in different directions. So one chemical will be moved inward, another chemical will be moved outward. So if you're in bio class, Bio 1, do you guys cover the sodium-potassium pump or talk about that at all? Mentioned briefly. All right. So the sodium-potassium pump is a classic example of an antiporter. All right. Now, with any of these transporters that we're talking about, all right, one thing that you should be aware of with them is that some of them are very, very specific for what they'll transport. For instance, the potassium channel, right? Excludes transports potassium. I think it's roughly 10,000 times faster than sodium, even though they're incredibly similar ions. Some transporters, though, are more promiscuous. So we've talked about a little bit about enzyme promiscuity with regards to evolution of protein function and stuff like that. Similarly, transporters can be promiscuous to a certain extent. They can accept similar types of compounds, allowing them to both basically to cross the membrane. So, for instance, there's a single transport. This is an example here for you. But there's a single transporter that will allow oxalate to cross the membrane, but also formate to cross the membrane. Right. So this is just one example for you. All right. Now, one last thing I want to talk about here. So when I've been talking about passive transport, what we've been talking about specifically is passive mediated transport, or facilitated, either mediated or facilitated, whatever. Facilitated transport. All right, meaning that these transport phenomena, they need a mediator. They need something to help them out, help the transport out. So you need a pore protein, basically, or a channel protein for these chemicals to cross the membrane. So whether we were talking about aquaporin, or water's crossing potassium channel, any of these different types of channel things, these are all examples of mediated transport. Right? It is also possible to do diffusion across a membrane in a non-mediated way. So there are certain chemicals that just by themselves can cross from one side of a cell membrane to another. Right? They don't need a channel or anything like that. All right, we call that non-mediated passive transport. All right, now, if we are, there's one very nice and easy way to distinguish between a mediated transport system and a non-mediated transport system. I would encourage you guys to pay really close attention to this right now, because this will come up again when we get to enzyme kinetics, and enzyme kinetics is hard enough as it is. So if you get this message now, hopefully it'll sink in that much easier later on. All right. So what we are looking at here on both of these graphs, the J, or on the example on the left, is basically the rate of transport. Right? It's something called flux, where flux is essentially the rate of transport. All right, so in either case here we have rate on the y-axis. Right? And these graphs are depicting rate as a function of the concentration of whatever is being transported. So these are plots of rate versus concentration of the whatever's being transported. All right, now the example on the left here, this is an example of mediated transport. The example on the right would be what we would expect for non-mediated transport. So mediated, again, is relying on a protein or some sort of a pore to get our molecule from one side to the other. Non-mediated means it can just, on its own, go through the membrane. All right, well, let's think real quick. We're going to do a little bit of kinetics from Gen Chem, low kinetics review. Right. Our reaction that we're doing here is just this molecule A, whatever A might be, from the inside of the membrane to the outside of the membrane, right? Right. If I was to write a rate law for this, all right, what would for just this simple reaction here? Right, we're going to ignore the back reaction right now. If I was to write a rate law for this, what would the rate law be? Anyone remember?
A we have with this superscript being governing whatever the balanced chemical formula is, its coefficient as an exponent over A to the inside.
Not quite. So you're close on it. So remember, rates of reactions are only in one direction. They don't depend on the product at all, right? They only depend on the reactant. So our rate expression will always be a rate constant K, right, times the concentration of our reactants to whatever power. All right, so in this case here, again, I'm only considering one direction transport. Our rate expression would be this. So the rate is equal to K times the concentration of the chemical, right? So this equation down here, if I made a plot of the rate versus concentration, what type of a curve am I going to get? Right? For this equation down here, if I plotted rate versus concentration, it's going to be a linear line with a slope basically showing you the rate constant. In this case, don't worry about calling it the P, but yeah, so the slope's K basically. So for a non-mediated transport, we basically get a linear response here, right? Now, the mediated case, this is the weird case, and this has a lot to do with enzyme kinetics and stuff too, right? What I want you to understand with this, I'm going to give you kind of an example, semi-real world example, is let's look first of all what's happening. So initially, this case is actually behaving like the non-mediated transport. So we increase the glucose concentration and the rate's increasing in more or less a linear manner, right? But eventually the rate starts to slow down and slow down and slow down, even though we continue to increase the concentration of the molecule. All right, so the real question is, why is it slowing down? All right, any ideas? Saturation is exactly it. So, one example for you, think of an Apple store, right? New iPhone came out, right? So, or, they say, you have 100 people in line at that Apple store, right? That store can only process so many people. It has a maximum rate that it can sell iPhones. Right? It doesn't matter if there's 100 people in line or 1,000 people in line. That line is only going to move at a certain speed, right? Well, let's say instead of 100 people, we have one person in line, right? Well, now the rate that they're selling iPhones is lower, right? If we have 2 people, all right, the rate's probably going to double. 3 people, it might triple. But once we get up to 100 people, now we've saturated the employees in the store. They can only sell iPhones so fast, right? So initially we see an increase in the rate until basically, in the biological example here, to switch to biological terms. So as we adding a little bit of ligand, it can go through. The glucose can get through faster. A little more glucose can get through faster still. All right, but we can reach a point where basically that pore is fully occupied with glucose. It can't let glucose through any faster than this maximal rate here, right? All right, so because of this, we get saturation. So we have saturation. We would also say that this point up here, this value, is the maximal rate. So the Y value right about there, that's the maximal rate. So if we went to infinite glucose concentration, we would be able to achieve this rate, right? All right, so those are just 2 ideas I want to get across to you, but for right now, the things I want you to know are that for mediated transport, they're going to tend to have curves like this that are going to saturate at this maximum rate, whereas for non-mediated transport, we're just going to get linear responses or linear correlation between rate and the substrate concentration. All right, now, talked about non-mediated transport a little bit. We have one slide on non-mediated transport, so this won't take long. All right, so how can molecules cross from one side of the membrane to another if there is no channel or no pore? All right, the answer is it has a lot to do with their polarity. Right? In general, the limiting factor for molecules being able to cross from one side of the membrane to another is how hydrophobic they are. Right? So first of all, they have to be polar enough that they're soluble in water. Right? If they're not soluble in water, they're never even going to get to the membrane because so they have to be at least somewhat soluble in water. But the trend here, so you can kind of see this upward trend with these different molecules, is that on the x-axis here, the more hydrophobic the molecule is, the faster it diffuses on the y-axis. Faster diffusion across the membrane. All right, so one really nice example of this, there are 3 of the chemicals on here we'll highlight because they're it's very easy to see this trend. All right, these 3 molecules up here, we have formamide, propionamide, and butaramide. All right, each one of these has an amide group at the end of it. All we're doing is we're increasing the length of the alkane tail. All right, so because of this, these molecules are getting progressively more hydrophobic. Right, and as they get more hydrophobic, they're also climbing up the permeability scale, so they're it's easier and easier for those molecules now to cross the cell membrane. So in general, assuming a molecule is soluble in water to begin with, in general, the more hydrophobic the molecule is, the faster it's going to be able to cross the membrane on its own. All right, so that is it for the passive transport portion of the chapter. So, we've got, we talked about a lot of different types of passive transport systems. The ionophores, which are basically antibiotics. They're molecules that can form temporary pores or carry ions across the cell membrane. These molecules tend to disrupt the membrane potential of a target organism. Or we have porins that are just straight up pore proteins that will allow certain molecules through. Right? We have ion channels that let ions of a certain size and certain charge to pass through. And we have other things as well, like the aquaporins that we talked about. We talked about the glucose transporters. Right? Ultimately, though, regardless of which one of these that we're talking about, we got this question here. What determines the direction of the solute movement? Question for you guys. So what determines which way the molecules are going to go in a passive transport system?
The chemical and electrical gradients.
Chemical and electrical gradients. So for a simple uncharged chemical, they will tend to move from areas of high to low, right? High to low concentration, right? When we were talking about charged species, then we also have to include the membrane potential, and things get a little more complex because of that. But we talked about those equations and stuff too. All right. So we also showed how some transporters, like say the potassium channel, has an open passageway all the time. Some, like the glucose transporter, undergo a conformational change that allows things to be transported. All right. Another example would be the voltage-gated channels. I guess with the voltage-gated channel, I'm going to add this in here. I forgot about it when I was going through. So I talked about how they open up due to that positively charged helix. I'm going to go back real quick because there's a detail I wanted to add in there. All right. So I talked about how they open up due to the movement of this positively charged helix. But I also told you that the part of the protein called the inactivation ball was really important. All right. Well, what happens with these proteins is that basically they open up due to a change in the membrane voltage, and they remain open for a very, very long time. What actually stops them from being open is there's a portion of the protein that's out here called the inactivation ball, and it's basically kind of moving around randomly, but eventually it'll find its way and insert itself into the pore, right, and basically plug up the pore until the conformational change occurs from open to closed. So it basically plugs a channel at a certain point in time until the channel itself can reclose. All right, that's the whole role of the inactivation ball. But back to this. All right, so the voltage-gated channels again require conformational change for their transport mechanism. And we talked about the different features that allow different types of channels, to discriminate between different types of ions. So we talked about how the potassium channel will allow just potassium to travel through because, again, it has weaker bonds to water, so the waters can be stripped off easier. But then the channel has a slightly bigger pore where it'll bind very well to potassium, but not to the smaller sodium because the bonds between the channel polar groups are basically too long for sodium to actually enter in there and bind effectively. We also talked about for water how it gets discriminated from hydronium, for instance, as it's traveling through that pore. All right, we'll just keep on going here. All right, so now we're on to active transporters. All right, so active transporters, these are transporters that will use energy of some form to basically induce transport of a molecule against its chemical gradient. All right, so in the case of, there's a classic example is the sodium-potassium pump. All right, So normally there's lots of sodium outside the cell, lots of potassium inside the cell. All right, the sodium potassium pump is going to move sodium ions from an area of low concentration to their area of high concentration. So it's going against that gradient. In order to do so, the system must be provided with energy. So a number of these systems, the energy source that they're going to use is ATP. All right, so that's one type of way that these pumps can get energy. All right, we'll talk about one of the very common types of these called the P-type ATPase pumps. All right, there's another family of transporters that are very, very important called the ABC transporters. All right, these are going to tend to move large organic molecules, for instance, like drugs, from one side of a membrane to another. All right, and then we're going to talk about a third class of active transporters. These ones do not use ATP at all. So these top two here, these rely on ATP and the energy from ATP. The bottom one does not. All right, the bottom one, in order to achieve active transport of one molecule, it allows another molecule to move down its chemical gradient. All right, so it's basically going to catalyze the favorable passive transport of one molecule and use that energy to cause the active transport of a second substance. All right, so the first type of pump we're going to be talking about are active transport system. The active transport systems, I will say, so anytime I'm talking about active transport, or let me say the converse way. If I say pump, all right, so-and-so pump, like the sodium potassium pump, all right, you immediately know this is an active transport system. So we're actually pumping something. We're using energy to move molecules, right? So the molecule or the protein that's shown here, this is a sodium potassium pump, or sodium potassium ATPase, as the book is going to call it. The ATPase basically says that it's going to hydrolyze ATP and use that energy to cause the transport of these ions. All right, so this particular protein, it has transmembrane domains here, which are going to form a pore to allow these ions to cross from one side to another. All right, but it's also got domains here in the cytosol. Right? These domains in the cytosol are where ATP is going to bind to the protein. And ATP binding and the hydrolysis of ATP are going to cause conformational changes in the protein that allow it to do this transport. All right. Now, the key to these type of pumps, right? The sodium potassium pump, or ATPase, is a member of a family of pumps called the P-type ATPases. All right, so this is just one example of a P-type ATPase. There are lots of other transporters that are also P-type ATPases. Now, what makes a P-type ATPase a P-type ATPase? All right. So the terminal phosphate on ATP. So I'm going to do rudimentary ATP here. So it's got the adenosine with 3 phosphates on it. All right. The end phosphate here is going to get cleaved off and transferred and form a covalent bond with an aspartate residue on the protein. Right? And what is going to form then is we're going to have a phosphorylated aspartate. Right? At one point during the mechanism. And this phosphorylation event is the key to unlocking the mechanism of these types of pumps. So what is going to happen is that when the protein is phosphorylated, this is going to cause a conformational change that is going to allow different events to occur. And when it's unphosphorylated, the protein will be in another state. So these P-type ATPases, they have 2 states, an unphosphorylated state and a phosphorylated state. Actually, real quickly, just to You know, I don't know, we've talked a lot of biology. I'm just going to give a little bit of chemistry here. Right. How does phosphorylation of an aspartate happen? All right. So here we have our ATP with the end phosphate on it. Right. For an aspartate, we have the carboxylate. Right. What's going to occur here is that you're going to end up with a nucleophilic attack of the carboxylate on the phosphate, on the phosphorus, right? And then we're going to break the bond here. Right? There's also going to this is the simplest simplistic I guess oversimplification of this. Let me do the full thing. So it'll bond first. We break actually the weakest bond, right? So the double bond gets broken and those electrons go up there, right? In a second step, right, I'll do the second step in a different color. Those electrons would come back down and then we break it, right? So in the end, we're going to end up with a phosphorylated aspartate and ADP as our other product. Right. Well, how does this happen? So how does this whole process work and actually lead to the pumping of sodium across one side and potassium to the other? Right. So initially, the protein, you know, it's sitting in the membrane here, and I'm just kind of draw like 3 little binding sites here. Right. It basically has binding sites for 3 sodium ions. Right. We call this the E1 state of the protein. That's just the name that we're going to give to it. Right, so this is the E1 state. The E1 state is capable of binding an ATP. So it can bind an ATP here. It can also bind the 3 sodiums. So the sodium I'm going to do is a little black circle here. So bind these 3 sodiums here. All right. And so we can end up basically with a complex. So we can start off with the E1 state bound to ATP. That can bind the 3 sodiums on the inside of the cell. I also want to point out this diagram is probably upside down to how you're used to thinking about membranes. So the inside of it is actually on the top, the outside's down below. All right, but it can bind these 3 sodiums inside the cell. And then that ATP can do the reaction we talked about. Right? It can transfer its phosphate to an aspartate of the protein. Right? And then causing ADP to be lost. So ADP goes away, right up here, right? And we now have a phosphorylated version of the protein with 3 sodiums bound, right? This phosphorylated E1 state does not exist for a very long time, right? Basically, as soon as the protein gets phosphorylated, it now turns into the E2 state, right? So the phosphorylated version is the E2 state. Now, the phosphorylated version of the protein, let's see how to draw this. Right. For the phosphorylated state, looks like this. So, in a cartoon diagram, right? So those 3 sodiums that bound initially, right? They basically, you know, they bound, and during the conformational change, a pore opened up, and they were able to travel through. All right. So the 3 sodiums are now on the other side of the membrane. All right. So they got transported over against their chemical gradient caused by this conformational change that was induced by phosphorylation of a single aspartate. All right. So those 3 sodiums are gone down here at the bottom. Right. They went away. All right. In this E2 phosphorylated state, right, the protein now has 2 binding sites inside that are optimal size for potassiums to come in and bind. All right, so these 2 potassiums, I'll draw them as kind of triangles, bigger triangles. Right, they can come bind to these 2 sites of the protein. All right, we basically get a complex between the phosphorylated E2 protein version of the protein, these 2 potassiums. And now the protein will actually undergo a second reaction that it can carry out on its own. So these proteins, the P-type ATPases, can phosphorylate an aspartate residue on them, but they can also then hydrolyze off that aspartate the phosphate from the aspartate. I'll show you how that works in just a second. It's basically the same chemistry we've been talking about. But as soon as that phosphate is removed down here at step 5, right, the protein no longer has its phosphate group on it, so it reverts to its E1 state. All right, this reversion now causes the potassium ions, channel basically opens up for them to cross over to the other side of the membrane, and we reset this whole process. Right, so that's basically what's going on throughout this mechanism. Now, just to go back here real quick, how can we hydrolyze off the phosphate? Well, I'm going to draw this slightly better than shown here, just to show all the atoms. All right, so we're doing a hydrolysis. So we want to use water to break a bond. Right. How can we make water more reactive in this? Anyone? Very easy. How can we, yeah, no. we're not gonna do redox or radical reactions here. What can we do with water to make it what type of reaction are we doing here? We're doing a nucleophile electrophile reaction. All hydrolysis reactions are nucleophile electrophile, right? Water, is it going to be our nucleophile or our electrophile? It's going to be our nucleophile, yep. So it's going to be our nucleophile because it's got the lone pairs of electrons on it, right? And we need to add water onto our molecules. We have to have it actually do some attacking here in order to do a hydrolysis. So the way we can make it a better nucleophile is to take a base and deprotonate it, right? All right, once it's deprotonated, now it can attack the phosphorus of the phosphate and push the electrons up onto the oxygen. Right. Then in a second step, I can get this to come up. All right, in the second step, the electrons on the oxygen, that alkoxide, will collapse back down, and we can cleave off the phosphate. All right, so in the end, in this step, we would end up with the free aspartate residue and a free phosphate. All right. So during that entire cycle, the net reaction of what's happening for the sodium-potassium pump are 3 sodiums get transported from the inside to the outside of the cell, 2 potassiums get transported from the outside to the inside, and an ATP gets converted into an ADP and a phosphate. Right, so it's not like, one of the take homes here I want to stress to you guys is, you guys all know that ATP is energy. You learned this in intro bio, right? How is ATP actually being harnessed? How is that energy being used? It's being used to cause conformational changes, and the way that that's happening is that N phosphate is getting added onto the protein temporarily. Right? This causes that conformational change. And then to reset the protein, that phosphate gets cleaved off. Right? And there's more we could say about the energetics of it, but I'm not going to go that deep with you guys right now. So if you have questions about that, let me know. All right. Anyways, that's it for today. Thank you guys for your time.